We already heard yesterday about the role of voltage-gated calcium channels in the afferent pain pathway from Diane. And so we really consider two types of calcium channels in afferent sensory neurons. The N-type channel, or CAF 2.2, which gates neurotransmitter release in dorsal horn synapses. And potentially also here in those nerve endings, as we learned yesterday, which is pretty cool. What I'm going to focus on is CAF 3.2, which is a member of the T-type calcium channel family. And what we know is that if we block these channels pharmacologically or we knock them down, we get analgesia across a wide range of preclinical models of either inflammatory or neuropathic pain. And conversely, if you cause a nerve injury, peripheral inflammation, chronic diabetes, gut inflammation, or even in post-surgical models, there is an upregulation of CAF 3.2 calcium channels in the dorsal root ganglia, as well as in the dorsal horn of the spinal cord, indicating that when you have more of these channels, you get more pain signaling. If you inhibit them, you get analgesia. And that is because these channels help regulate neurotransmission here, and they also regulate the excitability of the afferent fibers. Okay? So then the question is, why are these channels upregulated in all of these different types of insults? So it could be at the mRNA level, it could be lots of things. And what we did is we focused on a post-translational modification called ubiquitination. And so if you take the calcium channel sequence, you express it in hex cells, you immunoprecipitate the channels with an antibody against CAF 3.2, and then you probe for ubiquitin, you get a very beautiful band here, indicating that the channel is regulated by ubiquitination. And if you scan through the entire sequence of cytoplasmic regions of the channel, what you can find here in the domain 3,4 linker here, which has two splice isoforms, you find a proline tyrosine motif, which is a consensus motif for HEC-T ligase activity. And if you go ahead and you mutate that out, you lose some of this ubiquitin signal here, right? And so ubiquitination occurs at lysine residues typically. And so depending on the splice isoform, you either have 3 or 2 lysines in the 3,4 linker. And if you mutate this first lysine out, you have a very, very large decrease in ubiquitination. Now, there's a very nice poster out there by Henry Colecraft's lab on ubiquitination, where they overexpress different ligases and they see interesting modulation. And so keep in mind that this is basal ubiquitination in the absence of an overexpressed ligase. And it's quite possible that if you overexpress one of the HEC-T ligases, other regions beside the 3,4 linker might be important in the regulation of the channel. But we're just going to focus on this domain 3,4 linker here. Okay, now, we were interested in that potential sequence, and what we did is we made a biotinylated peptide. We pulled down a whole bunch of proteins from brain goo and isolated them, and we found a really interesting hit. And for the rest of my talk, I'm going to focus on that, and it's called USB5. And USB5 is a deubiquitinase, which does exactly the opposite of what a HEC-T ligase does. It removes ubiquitin from target proteins. There are about 100 dubs in the mammalian genome. And when we found USB5 and we started looking on PubMed, there were about 20 papers on it, none related to neuroscience. They were all about cancer. Okay? And so we got quite excited about that. And we had sort of a crazy hypothesis. And, this is one of the few times where a hypothesis actually holds up. We thought, what if there was an injury and suddenly you had more USB5 in the pain pathway? It would strip the channel of its ubiquitin groups, increase the stability, and then you have more T-type channels, and then you get more pain. Okay, so what we did is we wanted to make sure the interaction actually was real. So we did co-IPs from dorsal horn tissue and from dorsal root ganglion tissue. We could find an interaction, which is, of course, very important. And so here's an experiment where we've taken a mouse where we injected CFA into one paw. So this is basically causes a persistent inflammatory state. You have an ipsilateral and a contralateral paw, or you took a control saline, right? And then you isolate the dorsal root ganglia or the spinal cord tissue on the ipsi and contralateral side, and all you do is you probe for USB5. And what you can see is the DRG that are ipsilateral to the injection of CFA starts to produce a huge amount of USB5. And actually, consistent with our hypothesis for a change. And then we did this in spinal cord tissue as well. And here, this is just an ELISA that we've actually run from skin tissue. So even in the nerve endings in the skin, do we get more USB5? And this is going to be a little bit important later when we talk about translation, because that might be a biomarker for a type of chronic pain that we might go after when we make drugs and whatnot, okay? And we see this upregulation of USB5 in all of these different models that I've listed below. So it seems to be somewhat unified. Okay. Now, what happens when we knock it down? So we can inject shRNA against USB5 intrathecally, so which will knock down USB5 in the spinal cord as well as in the nerve endings that come in from the afferent fibers. Or you knock down an unrelated USB molecule. This is a formalin test where we inject formalin into the paw. There are two phases, an acute phase, which is basically characterized by licking and biting of the paw, lasts about a minute and a bit. And you can see that USB5 depletion shortens the first phase. And then the second phase is the inflammatory phase. And again, you have a very, very nice decrease in the duration of the nociceptive response, indicating that USB5 is important for the development of acute inflammatory pain. This is the CFA model, and here we're measuring simply mechanical withdrawal threshold in response to a stimulus that we apply to the paw. Normally that sits at around 9 grams, so that the animals are quite happy. When we give CFA, that threshold drops down to about 5 grams, so the animals have now allodynia and mechanical hyperalgesia. And when you inject shRNA, what you can see, you get a very, very nice reversal of this inflammatory pain phenotype that lasts about 3 days. Then the shRNA wears off, and then the pain sensitivity goes back down to the level that it was before shRNA. So that's now a chronic inflammatory test. And I should say we did a model of spared nerve injury here as well, and it works there too. Okay. So then we thought, okay, well, shRNA is not so practical. Can we just acutely uncouple USB5 from the calcium channel competitively? Now, we already know what the interaction site of USB5 on the calcium channel is based on the fact that we used it as a bait to find it, right? So we made a TAT peptide, so a cell-permeant peptide with about 25 amino acids stuck on there, which correspond to the interaction site. And we can deliver that either systemically or intrathecally to mice and ask the question as to whether or not that reverses inflammatory or neuropathic pain. And sure enough, it does, and it's actually remarkable. So here again, the CFA model, this is a neuropathic pain model. The mechanical threshold sits at around 9 or 10 grams. After CFA or nerve injury, it drops down. And then we give this TAT peptide, and within 45 minutes, which is very, very fast, we get a reversal of the pain phenotype, okay? And we can do this even 3 weeks after a nerve injury, and it still works, even though there's a lot of plasticity that occurs in the nervous system. Somehow, disrupting the interaction of USB5 with the calcium channel still produces analgesia, okay? And this works, again, systemically, and it works intrathecally. Systemically is very expensive because you need a ton of peptide, and it's not cheap to make. So most of the experiments that I'm going to show you are going to be done intrathecally. Then you can say, okay, well, you have a piece of the channel that's stuck to USB5, or maybe we're just blocking USB5 action globally and it can't interact with any target, and so it has nothing to do with the calcium channel. So there are two experiments that you can do to get around that. The first one is to figure out where on USB5 the calcium channel binds. And we did a lot of biochemical measurements, and we found that the CUBB domain of USB5 is responsible for interacting with the calcium channel. We chopped it up until we got about a 40-meric peptide. We put a TAT epitope on that, and there's a remarkable reversal of neuropathic pain that's actually complete when we applied that peptide. It was the same time course as we saw with the other peptide. So here we got a peptide against USB5. The idea is that it interacts with the calcium channel and prevents the USB5 from then binding to the channel. Okay, so now we have two complementary approaches, which give us confidence that what we're seeing is really specific. And then you can use a very nice trick, because their CV3.2 knockout animals have a compensatory mechanism that allows them to develop inflammatory pain, even though the CAF3.2 channel is gone. Okay, so that's great, because now you can give CFA, you get hypersensitivity, then you give the TAT peptide, and you ask the question whether it still works, and it has zero effect. And what that means now is that USB5 interactions with the calcium channel in the context of pain really require the channel to be there, and the peptide requires the channel, and it doesn't act on something else like a sodium channel or what have you. Okay, so that's all good. So then the question is, how can all these different types of peripheral insults all lead to the same outcome, meaning an increase in USB5 expression? And we thought, well, maybe it has nothing to do with the insult, but, well, it does, but maybe it doesn't matter what the insult is. Maybe all you do is triggering neuronal activity when you cause an injury, and then you have activity-dependent gene transcription or activation of USB5, and that's why it goes up in all of these different models, right? And so how do you test that? I mean, you can't ram an electrode into a mouse because then you would cause an injury. And so we used what Diane actually referred to yesterday very briefly. We used a transcutaneous non-invasive optogenetic approach where channel rhodopsin 2 is expressed in C fibers. And all you do is you take a laser or a diode, shine blue light onto the paw, and then you can trigger defensive response. And I'll show you what this looks like, right? This little mouse sits here, perfectly happy. Come on. Here we go. And the little laser goes on, and the mouse jumps away, right, and is unhappy about this. And you can show that this is not because of heat of the laser. It's really basically an activation of TRPV1 positive neurons that causes that response. And of course, that's cruel, and we're not going to be cruel to our animals. So what we can do is we can put them under anesthesia and then stimulate them for just 10 minutes at a certain power level, at a certain type of, at a certain frequency. And then either we wake them up and we behaviorally assess them, or we sacrifice the animals an hour later and ask whether optogenetically induced neuronal activity led to an increase in USB5 expression or not. And that's exactly shown here. And so we thought, well, isn't that really cool, right? So you trigger activity-dependent expression of USB5, T channels become deubiquitinated, they contribute more to firing of the neurons, and this goes on and on and on, and you have this self-perpetuating loop, and when we disrupt the interaction, it stops, right? So here's a hypothesis that absolutely failed, because when you look at behavioral sensitization, when you wake the animals up, an hour after you wake them up, they have a reduced threshold for mechanical and thermal stimuli, but after 24 hours, that spontaneously disappears. And if you do a biochemical measurement right at that point, USB5 levels have gone back down. So it's not self-perpetuating, okay? And so, I'm going to skip this, but so what we have is we have a scenario where you cause a nerve injury or peripheral inflammation, USB5 is upregulated, it removes ubiquitin from the channel, you end up with more channels and you have persistent pain, right? You do optogenetic activation, exactly the same thing happens, but you have a transient pain state. So how can that be? So there is clearly a cofactor missing here that allows this to be transient as opposed to persistent, okay? And so we have some evidence that I'll go over very quickly that might point in the right direction, and that suggests the innate immune system may be involved in this. And so when we inject CFA into the hind paw and we look for upregulation of many different things that are associated with innate immunity, we see an upregulation of a number of toll-like receptors, particularly TLR2 and TLR6, which form heterodimers, okay? And so what we found was that when we took TLR2 knockout animals and we gave them CFA, there was no pain phenotype. Okay, so clearly TLR2 signaling is important to give you CFA-induced hypersensitivity. And then what you can do is you can look for USB5 upregulation in wild-type animals or TLR2 knockout animals. And while you get an upregulation in the wild-type animals with CFA of USB5, right, in the TLR2 knockout animals, this doesn't happen, indicating somehow toll-like receptor signaling is involved, right? And so toll-like receptors do lots of things. But we zeroed in based on some experiments that we did on interleukin-33. And what we find is that after CFA, in DRGs or spinal cord, or even in the hind paw, we get an upregulation of interleukin-33 levels, okay? And so the question then is this important? And there happens to be a receptor for interleukin-33, which is called ST2. And if we take a functional antibody against ST2 receptors and inject them into the intrathecally, okay, you completely block thermal hypersensitivity that's induced by CFA. And I don't have time to show you all of this, but what we have here is basically a model where we have a two-step process, right? There's an injury, so such as CFA. You get TLR receptor activation, IL-33 is being produced, and somehow nerve endings are being primed, okay? And then there's a secondary infiltration of macrophages to the DRG, and they release IL-33, and there are there are ST2 receptors directly on the DRG neurons. And so if you were to go ahead and just inject IL-33 peripherally, you get a transient pain response. If you inject it intrathecally, you get a transient pain response. But if you do it at both sites, you get a huge potentiation, and it becomes a very much more long-lasting effect. So we think somehow USB5 plays a role in that. We haven't worked it all out yet, but at least we have a working hypothesis where we can try to dissect the pathway and figure out what's going on. So then we thought, well, can we maybe weaponize this interaction for the purpose of therapeutics, okay? And so we developed a high-throughput assay where we have a biotinylated peptide containing the 3.234 linker, immobilized, and we take recombinant USB5 and we can detect this interaction with an antibody, okay, so it becomes fluorescent. And then we can take molecules from compound libraries and put them into the dish and ask the question as to whether or not the interaction is disrupted, just like we did with the peptides, okay? And so we now screen tens to hundreds of thousands of compounds, but here's just an example of one compound series where we found these molecules here that have this sort of rhodonine group attached to an amino acid-like structure, right? And there's a series of 11 molecules that we looked at as part of this particular subseries. And what we find is that the first 8 here are very, very potent in disrupting the interaction of USB5 with the calcium channel. And the remaining 3 ones that are down here have no effect whatsoever. So indicating there is a structure-activity relationship that's present in that particular compound series. You take the first one here, which we call, I don't know why we called it I-1. I think it was the patent attorney. Anyway, so we called it II1, right? And we do a dose response curve and we get about a 1 micromolar AC50, okay? And then what we can do is we can do a modeling where we take the crystal structure of USB5, this is getting weak here, and we dock the domain 3,4 linker onto there. So this is done in silico. So there's a helix in the 3,4 linker that really nestles very nicely here where the CUBP domain is, right? And that was the one that we found biochemically as well. And then what we can do is we can in silico dock the entire compound series. And what we do is we find 2 sites, one on the bottom here, that all the molecules dock to in a somewhat similar orientation. And up here, you got 8 that dock in one way, and the 3 that are inactive point in a different direction, which might maybe explain why they're not effective in disrupting the interaction of the calcium channel with these particular molecules. But what we can say is that these molecules are very, very effective in inflammatory pain. So this is the formalin test. There's a dose-dependent reduction in both phases. And this is the CFA model where we get complete reversal of inflammatory pain. And once again, if we use CAV 3.2 null animals, these molecules have no effect, indicating you require the calcium channel for them to do stuff and they're not acting on something else, right? And keep in mind, the beauty with this is now that you have a process that's only turned on, at least in peripheral neurons, when you have an insult. The molecules don't block the calcium channels directly. They just prevent an aberrant upregulation of the calcium channel that happens in a pathological process. And so when we then target that, there's much reduced potential for side effects, at least in theory, okay? And I should say that we have second-generation compounds that work really, really well. There are now 100 nanomolar affinity, a different compound series from what I've showed you before. And that's what we're going forward with the company that we started, which Raj mentioned. It's called Cymodan Therapeutics. Anyway, the mechanism basically is that these molecules simply disrupt the association of USB5 with the calcium channel, thereby preventing deubiquitination, and then you have fewer channels and you get less pain. Okay, and I'm going to show you 3 more slides and then I will shut up. The question is, of course, mice are fine, rats are fine, but what happens in humans, right? And so, not fortuitously, because it's awful for the patient, but there was we were contacted by neurologists, pediatric neurologists from Oslo, and they have this patient that has a congenital insensitivity to pain. So at the time, it was an 11-year-old boy, slight developmental delay, mostly normal, but has chewed up fingers from self-mutilation because this individual does not feel any acute pain. And the geneticists sequenced the whole genome, and the only thing they could find was a single point mutation in position 24 of USP5. So we were contacted by these neurologists and say, well, what's going on here? And so we looked a little bit, and what we find is that when you take wild-type USP5 or you put the mutation in there is no effect on the catalytic activity of USP5, okay? So it still acts as a deubiquitinase. But what we find is that it becomes more ubiquitinated itself. So USP5 has a ubiquitination site on it, and where it can be self-ubiquitinated, and therefore, well, it's not self-ubiquitinated, it can be ubiquitinated, so the protein levels of USP5 can be regulated, and that's increased, and then the prediction would be that you would have less protein around, okay? And we got sick and tired of playing around with biochemistry and overexpressing viral constructs in animals, so we decided to just make a knock-in mouse, so we used CRISPR, and we knocked the mutation in, and what we find here is, so we give CFA, okay? Normally we get a nice behavioral sensitization to thermal stimuli or mechanical stimuli, right? So you've seen this before in other slides. In the mutant, that is dramatically attenuated, if not completely absent. Statistically it's absent, but there is a bit of a trend here, right? Indicating that these animals are now resistant to CFA-induced inflammatory pain. And if you look carefully, you can see an upregulation in wild type, okay, in the DRG, when you give CFA into one hind paw. In the mutant animals, there's much, much less there. And as a result, you have more T-type channels here compared to the mutant. Okay, so it kind of all fits together. And then this is even cooler. This is the basically a neuropathic pain model, okay, where we're again looking at mechanical and thermal thresholds. Normally, you get behavioral sensitization after a nerve injury. In the R24 mutant, you don't have that difference anymore. So these animals are resistant largely to inflammatory pain and neuropathic pain, which phenocopies what you see in the patient, okay? And so for us, this is important because this is the first evidence that USP5 may be important somehow in regulating nociception, at least, in humans, okay? Now, the idea then is to come back and say, okay, let's say you had a cohort of diabetic neuropathy patients, half of whom may develop neuropathic pain, the others don't. You take a skin biopsy, you look and see whether or not USP5 has gone up. And if you ever get to a clinical trial with any of the molecules that we're trying to develop, you'd want to go after the patient population that shows elevated USP5 compared to those that don't, right? We're far away from that, but hopefully one day we'll get there. So what I showed you was that if you give all sorts of different insults, you get an upregulation of USP5, which is a deubiquitinase. Because it deubiquitinates COP3.2 channels, you end up with more of these T-type channels in the membrane, and therefore you have more pain. And if you interfere with this interaction, either by knocking down USP5 or using a TAT peptide, this competitively inhibits the interaction, or a small organic molecule, then you get analgesia, okay? And in some ways, this is just the correlate, the calcium channel correlate to Raj's beautiful sodium channel story, where you have a regulator that happens to be more complicatedly regulated by stimulation and ubiquitination. And again, you're interfering with the regulatory pathway, right? And you affect hypersensitivity in a number of preclinical and possibly clinical models. So it's actually a kind of a nice approach and very nice parallel. And of course, I do nothing but write papers and grants. So the people that did the work, Vinicius did most of the behavioral studies that I showed you. Agustin discovered USP5 as a binding partner and is doing a lot of the biochemistry. Ali did the modeling that I showed you, and Eda contributed as well, along with Chunting on the IL-33 project. And with that, I will be done. Thank you. I'm sorry, I don't have Dick Chen
Thank
jokes.
you, Gerald. Fantastic story. Arden, you had a question? No? Henry. Or, go ahead.
Me? Hi. Great talk. Thank you. That's cool. Did I notice in your IL compound, the CFA experiment, there's some desensitization over time to the IL compound?
No, I don't think so. Well, okay, hang on. Not desensitization, I think it just wears off. Hang on, I'll go back, okay?
So it's just cleared or metabolized over time?
everything wears off to this, right? So this particular molecule lasts for about 90 minutes, 180 minutes, and after 270 minutes, it stops working, right? So that's that particular molecule. With a single dose, will last that long.
Okay. And then related question, with the 3.2 knockout mice, they develop some inflammatory pain phenotype. Is there any potential for long-term dosing of these molecules to induce the same kind of remodeling? Is that possible?
So everything's possible. Of course, a knockout animal is very different from something that you knock down acutely, right? Because there's all these compensatory processes during development. So I would not jump to the conclusion that acute inhibition for a prolonged period of time will do the same thing as genetic knockout at an embryonic stage. But you'd want to, unlike Raj, who had these nice repeat data on dosing data for NAV 1.7, we haven't done that yet. And we will certainly look at that. But we don't expect the same kind of tolerance that you would see with, for example, an opioid, right? Because it's a totally different pathway in terms of the way you develop tolerance.
Oh, back here. Very interesting talk. I have two questions. One is, could you comment on the source of the IL-33? Presumably it's some type of an immune cell, perhaps. And the second question is, do you see any differences between male and female mice in your paradigms, given that there's a lot of evidence for, especially inflammatory T cell or microglial-mediated sexual dimorphism?
Yeah, so with both the IL-33, so the second question first, because it's the easy one. We tested that in female animals, and the IL-3 project worked beautifully in female animals, and the USB-5 approach also works beautifully in female animals with slightly different kinetics, but subtle. But that's important because, from it doubles your market. And of course, you don't want to have a process that's completely affected by the sex of the animals or the patient. The IL-33, so what we know is, and this is published in Cell Reports in 2021 or something like that, I can't remember. What we see is that you have a primary immune cell infiltration into the peripheral tissue, including macrophages. And IL-33 is being produced, but IL-33 receptors are not present on nerve endings in the skin. There are skin cells that have the receptor, and IL-33 interacts with that, and then there is another soluble messenger that we don't know what it is that seems to act on the nerve endings directly, okay? And we know that because the nerve endings start to fire more. At the level of the spinal cord and DRG, we see macrophage infiltration after CFA, okay? We can detect that. We see an upregulation of IL-33, but here the IL-33 receptors are actually on the neurons now. And so when we give an intrathecal functional antibody against the IL-33 receptor, we get analgesia. And that analgesia works in CFA and in neuropathic pain models, even though the first one requires TLR2, but the second one does not. Okay, so there is some convergence of pathways to IL-33, but it's a lot more work to be done. So I'm trying to kind of be fuzzy about it because I don't know the answer.
Hang on one sec. Sorry, Henry. Training question first.
Okay.
It's me. Hi, Dr. Zamponi. Thanks for your talk. It was great. Have you ever tested if this regulation by USP5 is selective for CAV 3.2?
So we looked at interactions with 3.1 and 3.3, and off the top of my head, it works on one of them, but not the other. Okay, but we didn't do any functional studies on it. But there is a potential that it interacts with them. In the afferent pain pathway, there is a little 3.1 and there's a little 3.3, depending on which neurons you're looking at. So it could play a role there. But because the 3.2 knockout animals are so resistant to any manipulations that we do, we're pretty confident that most of the actions that we observe are going through 3.2.
Last question, Henry.
Great. So beautiful talk and really fascinating data. Thanks for that. I had a question about the young man with the sensitivity to pain.
Yeah.
Is he homozygous or heterozygous?
He's hetero.
Hetero.
Yeah, and when we make a homozygous mouse, most of them don't survive. And if you knock out USB5 completely, it's embryonic, mostly lethal. So you need it, but if you have one copy, then you're already compromised. Like one compromised copy, you're in trouble.
All right, thank you. That was a great talk. Thank you very much.
Thank you.